Good morning, everyone. I'm truly grateful for the opportunity to speak with you today, albeit remotely. Unfortunately, due to a scheduling conflict, I'm unable to join you in person this time. I hope to have the chance to meet and engage with all of you in person at future events. So for today's talk, I will talk about my recent research on the novel computational method for accurate and efficient integrative reference-informed spatial domain detection for spatial transcriptomics. As we have all learned from the previous two speakers' wonderful presentation, they gave a very comprehensive overview about the technologies part of the spatial resolved transcriptomics. And briefly, spatially resolved transcriptomics is a set of new technology that currently revolutionize our understanding of biology today. And actually, here is a image. Here is a illustration to show you the differences between these three transcriptomics technologies. So before the spatial transcriptomics, the bulk RNA-seq is the traditional way to measure the gene expression, and which actually measures the average gene expression of a sample. However, we don't know what the cell types are in this sample due to the technical limitations. So here is the analogy of this sequencing technology. And we can imagine that it is like a painting with the mixture of dots of colors, but we actually don't know what the exact color is for each dot. And then the single cell RNA sequencing technologies become popular. And different from the traditional bulk RNA-seq, it is able to first isolate cells and then measure the gene expression profile for each individual cell. So we can also use this color palette as the analogy. And now we know the exact color of each dot is, and the color tone can corresponding to the different major cell types. And within the same color tones, the dots of colors can aggregate it into the same color tone. And they also show some heterogeneity, which can indicate the cellular heterogeneity can corresponding to their different cell states or cell type populations. However, the single-cell RNA sequencing technology cannot capture the spatial information of cells. So this leads to the failure in the understanding of the organization of cells in the tissue landscape. Hence, the spatial transcriptomics has been developed over the last decade. And analogically, it can be viewed as a pointillistic painting where the dots of colors applied in patterns to form this kind of image. So now we know the exact position of each dot in this kind of painting with mixture of dots. But for most of the sequencing-based technologies, each spatial location actually contains a mixture of the cells as introduced by the previous presenter. And so here is a simple illustration of what spatial transcriptomics is. And briefly, the spatially resolved transcriptomic technologies has enabled the gene expression profiling with spatial localization information on the biological tissues. And there are several spatial transcriptomics technologies available. And one of the most popular commercially platform is the 10x Visium. And here is a simple illustration of how this platform generates this kind of spatial gene expression data. So first, you will have a commercially available slide with about different squares from this company. And each square containing about 5,000 barcoded props containing the spots of barcoded mRNA props. And then you overlay a specific tissue on the top of the squares, and the mRNAs released from the tissue will be hybridized with the barcoded capture props underneath the tissue. And then these props will allow you to capture the gene expression information. And because these barcoded spots also contain the spatial barcode, so you will also obtain the spatial information. So at the end of the day, through these spatial transcriptomics technologies, you will get a set of two data matrices. Specifically, one is this N by P count matrix of the gene expression measurements, where the N is the number of spatial locations, and the P is the number of total genes. And another data matrix we have is the spatial location matrix with usually two-dimensional coordinates that represents the spatial relationship across all these locations. We call it spot here. Once we have this kind of data, actually for these 10x Visium sequencing-based technologies, there are several challenges in this kind of sequencing-based technologies due to the technical limitations. So the first challenge is that because the transcripts captured at each spot actually contains a mixture of cells, and actually the cell type composition in each measured spatial location is unknown. We only observed that count data from the data we obtained. So we previously developed a method called CART to perform this spatially informed cell type deconvolution for spatial transcriptomics. So the deconvolution means that this method CART can estimate the cell type proportions for each spatial location. And once we get these vectorial proportions for each location, and we can jointly visualize them to see what are the regions that have a high enrichment of some cell types, or even see if there are some cell types co-localized together spatially. So as a methodology developer, I'm always curious how my method can be widely used to broad systems of the biological context. So previously, we found that a very nice benchmarking paper published on Nature Communications, they benchmark 18 methods for the integration of the single-cell RNA-seq and also the spatial transcriptomics. And in terms of the performance, these CART, cell-to-location, and tangram are the top 3 best methods for conducting the cellular deconvolution task. And this figure summarizes all the metrics they evaluated in terms of the accuracy, the robustness to different scenarios, and also the usability. And the darker color and representing the best rank that this method achieves. So we are glad that CART has been widely used for addressing this kind of challenging that the cell type composition is unknown. And actually, CART has been widely applied in different tissues. For example, we apply it on this mouse olfactory bulb tissue to estimate the cell type composition, which reflects this layered structure of this mouse olfactory bulb tissue. And we also identify this intratumor heterogeneity to identify 2 specific regions that are enriched by different cancer clone cell types on this pancreatic ductal adenocarcinoma tissue. And we also apply CART on this hippocampus high resolution slide 6.2 version data, which our CART is the method that can identify clearly these 3 CA1, CA3, and dentate gyrus subregions of the hippocampus. And also, we found that for some other people in the field who generate their own spatial resolved transcriptomics data, they use CART to estimate the cell type compositions. So it has been widely applied in various tissues. For example, a paper published on Nature Cardiovascular Research, they use CART to estimate the proportion score of the cardiomyocyte, to see whether this proportion score changes from this control tissue to day 1 to day 14 in the marine hearts tissue. And also a recent paper published on Nature Immunology, they used the CART to estimate the cell type proportions by computing the changes in the abundance of about 195 cell types across all these 9 organs, which here they show these liver, kidney, and the clone tissues, and CART can provide novel biological insight in terms of the cellular heterogeneity on their spatial distribution. And in terms of this heart tissue and also the whole organs, CART has also been widely applied in various cancer tissues. For example, they apply CART in the melanoma, published a study in the Nature Communications, and also the other examples. And finally, CART is also computationally fast and memory efficient. And it's compared with all the other methods, it only takes about 10 seconds to finish, 10 seconds to 6 seconds to finish all this PDAC tissue, and only requires 300 megabytes. So for the previous slides, I introduced how computational method CART can address the first challenging about the unknown cell type compositions. And CART can also do the high resolution imputation as well as the spatial mapping of the single cell resolution based expression. For more details, you can check our Nature Biotech paper. And Another essential task of the spatial resolved transcriptomic studies is to characterize the spatial organization of complex tissues in the form of spatial domain detection. And detecting spatial domains on the tissue in these spatial resolved transcriptomic studies can facilitate our understanding of the spatial and functional organization of normal tissue and reveal how alternations in the tissue structure may underlie disease etiology. Actually, for this task, several methods have been developed to detect the spatial domains, such as the SPAR, GCN, Bayes space, and all the other some deep learning methods. However, there are several limitations for these methods. First, almost all of them, most of them analyze one tissue slice at a time and does not handle multiple slice experiments, especially nowadays the data are accumulating at more multi-sample scale. And second, there are still large, because the spatial domains actually represent the spatial organized and functionally distinct anatomical structures on the tissue that are characterized by their varying cell type compositions. However, all these existing methods directly rely on the transcriptomic heterogeneity to disentangle the spatial domains, and such transcriptomic heterogeneity across these spatial domains is only actually a secondary feature of the spatial domains, as it is a direct consequence of the unique cell type composition underlying each spatial domains. Thus, there is a large room for the improvement in terms of the performance on the domain detection accuracy, as well as the interpretability on the domains they detected. Finally, all of these previous methods are designed for data sets with low to moderate size. They are not computationally expensive to high dimensional data sets with more than 100K spatial locations like the 10x Xenium for even one slice. And nowadays, people generate multiple slices at this scale. And I think so all these motivations and limitations motivate me to develop a novel method, which is called IRIS. And IRIS is short for Integrative and Reference-Informed Domain Detection for Spatial Transcriptomics. And here are the key idea and rationale behind this method, which actually correspondingly to address all the previous limitations. First of all, it is a reference-informed method that allows us to take advantage of this vast amount of the data collected in the parallel research field of the single-cell RNA-seq studies and integrate these invaluable cell type specific transcriptomic profiles for the improving the domain detection accuracy as well as the interpretation on these domains we detected. And integrating this information will potentially improve the performance. And second, it is an integrative method that jointly analyzes multiple tissue slices, both adjacent or non-adjacent, and it can scalable to hundreds of slices in our recent evaluations. And third, it is very computationally efficient and applicable to very large-scale spatial resolved transcriptomics data sets with millions of spatial locations. So before I introduce the methodology details, I'd like to first introduce the main modeling assumptions to get an idea of the rationale behind this method. Specifically, we assume that each spatial domain is characterized by their unique composition of cell types. And then for a given spatial domain, across these slices, if the locations on different slices belong to the same spatial domain, this domain is characterized by their cell type compositions that are consistent across these slices, but we still allow for the variability in terms of their cell type composition or low dimensional representation in the cell type space to allow a variability. But we assume that their mean is consistent across these slices, across all for the same domain. And the third assumption is that within the same slice, the cell type compositions share high similarity between their neighboring locations to encouraging the within slice similarity in terms of their spatial locations and similarities. So let's look at the pipeline of our method IRIS first. IRIS requires two types of the data input for the reference-based version. The first input is the single cell reference data that serves as a reference and to provide the cell type specific information. And the second one is a list of spatial transcriptomics data set that contains these transcriptomic profiles from multiple tissue slices. These slices can be adjacent or non-adjacent. And then IRIS builds upon the fact that multiple slices from the same tissues often contain a similar set of spatial domains characterized by their similar cell type compositions, and their neighboring locations within the same tissue usually share high similarities. So in this non-active matrix factorization framework, coupled by their within slice similarity penalty and also between slice similarity penalty, IRIS accommodates this cell type compositional similarity across locations within the same slice, and also this mean cell type compositional similarity across slices for the same domain to borrow information both within and between tissue slices. And by encouraging these two similarities, IRIS first updated the low-dimensional embeddings in terms of the cell type compositional matrices, and then concatenate them into a concatenated matrix. And then we directly perform unsupervised clustering algorithm, for example, the k-means, just k-means algorithm on these concatenated matrix to update the spatial domains for each iteration. And then we iteratively update the composition matrix, concatenated matrix, and also the spatial domains to achieve the optimal performance. And for this method, we have examined 7 real spatial transcriptomics data sets from all the popular technologies from different tissue and different technologies, including the 10x Visium, SliceSeq version 2, and also the Cosmics, and also the 10x Xenium, and also the VisGen mouse Murphish, and also the StereoSeq. And here I will use two real data as a motivating example to show to demonstrate the accuracy of our method and robustness of our methods. The first data set I will use is a commonly used benchmarking data set as an example to show how accurate IRIS is. So this data set is a widely used human dorsolateral prefrontal cortex data, which contains these manual annotations from the biologists from this original publication that can serve as a ground truth for us to evaluate the accuracy by comparing different methods. So we first perform the analysis on the baseline setting where the tissue slices that are from the same donor that represents a more homogeneous setting. And I compare IRIS with the current methods. So the first column represents the manual annotation that can be served as a ground truth for us to measure the domain detection accuracy. And the second column is our results domain for detected domains. And the first group of the method that we compare is this single slice-based method, because we want to see whether the model really benefits from jointly modeling them. So this method, including this the Heidelmarkov random field based method, Bayes space, and the variational autoencoder based method as SEDR, and the graph convolution neural method based SPAR GCN, and also contrastive learning based method graph ST, and this GAE method Stargate. And we found that all these IRIS substantially performs better than all these methods, which indicates the benefits of modeling multiple tissue slices. And then we were thinking about whether IRIS can take the benefits from the iterative updating for both low-dimensional embeddings in terms of the cell type composition as well as these domains. So we compare the second categories of the methods where we perform a naive combination of, first, we perform the deconvolution by using the CART or RCTD for each slice, and then we concatenate them together, and then following the same procedure for IRIS. And we found that this symbol combination, two-step based methods performs not good as IRIS, which indicates the benefits of the joint modeling multiple slices as well as iterative updating both results in our modeling framework. And then we also, because after we finished this method, we also saw some methods that focusing on multi-sample domain detection. So we also added this comparison to really show what, the benefits of IRIS. So the method we compare is the BAS, which is a Bayesian method that can incorporate multiple tissue slices, but their computational burden is a lot, so it cannot deal with the recently released high-dimensional data such as the 10x And even for this, we compare with also other deep learning-based methods that can extend their framework into multiple slices. And we found that still IRIS substantially performs better than all the other methods, which indicates that the benefits for integrating the reference-based information to facilitate this domain detection. So all these previous methods actually compares focusing on external methods, and it can be due to the modeling implementation. So to ensure the rigorous of the benefits of IRIS modeling framework, I also implemented different variants of IRIS within the same modeling framework. So we compare the single slice version of IRIS and also the reference-free slice version of IRIS. We found so in the single slice, we use IRIS to analyze one slice at a time. And in the reference-free, we don't utilize this single-cell reference. And we can see that the IRIS can still, all these three versions of IRIS performs very good, even compared with the previous categories of methods. And the key point for this figure is that for the baseline setting, the benefits is more from this reference information. While for the challenging setting, jointly modeling the multiple slices and incorporating the reference information will substantially improve the results. And these are the quantification to show that the IRIS is more accurate than all the other methods. The larger of the adjusted rand index represents the more consistency with the manual annotation. And we also perform the analysis, evaluate the robustness of our method on different challenging settings. So the first challenging setting is that the slices from different donors, where it is more heterogeneous, and IRIS substantially performs better than all the other methods. And the second and three challenging setting is to see how IRIS is robust to these to the reference single cell information. So we evaluate if IRIS can still performs better in the in the situation when there are missing cell type in the reference data, and also even the misclassified cell type in the reference data. And we found that IRIS still substantially performs better than all the other methods. And finally, we also think about if we utilize another external single-cell reference data, because sometimes users might feel challenging in selecting the reference data. So we hope our method can be robust to a various set of reference data. And this data, we can see that their resolution is different, and also the technology generate the single-cell reference data is different. And still, IRIS substantially performs better, which indicates the robustness of our methods to the reference choice. And finally, we also find that our detected domains are more biological and meaningful. So we checked the domain-specific layer genes, the higher fold change for that layer-specific genes in terms of each domains we detected for different methods, which indicates that it is more matched with the biological meaning. And we found that for all the domains, our IRIS detected the domain-specific, the genes has a higher fold change expression in these domains comparing with all the other domains. And the second data is we also apply IRIS on this 10x data, which is public available human breast cancer 10x data. And only IRIS can run analysis on this large-scale data set, which contains more than 100K spatial locations jointly across 2 slices. And the breast cancer is a heterogeneous disease with high intratumoral and intertumoral variation in the histological and molecular features. And the invasive ductal carcinoma and ductal carcinoma in situ are 2 most common forms of the breast cancer, while the invasive ductal carcinoma is more aggressive than these DCAS. And a substantial proportion of the patients with the IDC have accompanying DCAS component. And we want to see if IRIS can identify these intratumor heterogeneity to identify these different DCIS and IDC regions. So here are the results of IRIS. We show the domains we detected. We can see that IRIS can clearly identify 4 distinct tumor domains, including these 2 DCIS domains that including non-invasive forms of the breast cancer, and 2 IDC domain, along with other domains belong to the part of the tumor microenvironment, such as the immune-related domains and tumor stroma regions. Specifically, the domain 5 and domain 8 represent the invasive ductal carcinoma region, while the domain 10 and domain 16 represents this DCIS region. And we further checked the marker genes. For example, we found that this TCM is a candidate breast cancer oncogene associated with beta-catenin pathway that elevates this invasive behavior of the cancer cells. And we found that it is highly enriched in the domains 5 and domain 8, which is actually corresponding to these invasive ductal carcinoma regions. Well, this CEACM gene 6 gene is a marker with a high expression in these ductal carcinoma in situ regions, and we found that its enrichment is higher in the domain 10 and domain 16, corresponding to these ductal carcinoma in situ regions, and much higher than these invasive regions. And we also carefully examined the hormone receptor status of these tumor regions to characterize the invasiveness and this intratumor heterogeneity of the tissues. We found that this epidermal growth factor receptor ERBB2 is highly expressed in these invasive ductal carcinoma domains 8 and 5, more so than that in these ductal carcinoma in situ regions 10 and 16. And we also found that this hormone receptor ESR1 is mainly expressed in these IDC and both DCIS regions, while this hormone receptor PGR is lowly expressed in all domains, which indicate that this breast cancer patient might be the HER2 positive, ER positive, and PR negative clinical type. And we also quantified a lot of the proportions of the spatial locations in each domain. We found that it displays a high expression of the tumor invasiveness marker genes, and further confirmed that the domain 8 is more invasive than domain 5 and 10 and 16. Specifically, the AGR3 gene is associated with low histological grade breast tumors, and the proportion of spatial locations marked with the high expression of this gene is higher in the ductal carcinoma in situ regions. However, the CENPF is associated with tumor aggressive features and poor prognosis of patients with breast cancer. And we found that the proportion of spatial locations marked with this high expression of this gene is higher in the invasive ductal carcinoma regions, domains 8 and domains 5. And finally, IRIS is computationally fast and memory efficient. For the moderate to high dimensional data, it's about 4 to 100 times faster than the existing methods while using only like average 30% of the physical memory required by this method. And for the Salem data, we only take about 20 minutes to finish under the CPU resources. And recently, we tested our collaborators' data sets on like more than on 200 slices, and it only takes about 20 minutes, even on the CPU resources. So to summarize, IRIS is a highly scalable method for integrative and reference-informed domain detection for spatial resolved transcriptomics, and it models multiple tissue slices jointly, produce highly accurate and interpretable results, and is computationally efficient. And in real data analysis with different tissues, here we show the human DLPFC, which the manual annotation is available. It currently captures the layer structure in both baseline and challenging settings with very high accuracy that was not achieved by all the current methods. And it will, we hope that it will serve as a powerful tool for large-scale spatial transcriptomics tumor data sets to study the tumor microenvironment and review these heterogeneity. But it's robust to be applicable to a wide range of the tissue settings. And the paper is accepted by Nature Methods, and we hope to see it online soon. And thank you so much for your attention, and any comments and suggestions. Also, the collaborations are welcome, as I just started my lab at the Brown Biostat Department and also the Center for Computational Molecular Biology. Thank you so much.
Hi, can you hear me?
Yes, yes.
Great. Thanks for the beautiful talk. I really love that method. I just sent it to my student from your GitHub page. So one of the things that I think a lot about as a biologist, right, who's interested in different tissue spaces are the cells that you can't get out by single cell, right? So a lot of us are working in tissues where you mash them up you get some of the cell types, but you don't get most of them, like stromal cells, for example. And that's a loss in the single cell data set. So when thinking about the reference single cell data set, did you really, I'm curious if you guys considered sort of different data sets to use and how you make the best reference data set so that you're capturing that information spatially when you do these associations of the single cell to the spatial data.
Yeah, thank you. That's a great question. So the single cell reference, we utilized that we have evaluated. So from the methodology perspective, we evaluated our method's accuracy in terms of like when we create, like, for example, if there are missing cell types in the single cell that corresponding to the setting, when you said that some cell types cannot be captured, and our method is still accurate and robust. And we also evaluate the setting when there are cell types that are misclassified which represents the setting when the cell type annotation is uncertain, and our method still performs better. And I think we also for the data set where we evaluate the accuracy, we also evaluate different settings of the reference information. And actually, in this tutorial, we also released a reference-free version, which can allow a flexibility about the number of cell types were in the reference data. But I think for the future plan, it's better to probably formulate this work into multiple references framework that can facilitate the selection of the optimal references. And I think that's more for the future step. And I think it's a good plan that if we can incorporate the multiple slices and at the same time to select the best representative references for each spatial transcriptomics data. But our target is more focusing on detecting the domains that can be characterized by the current cell types existing in the reference that based on the prior biological knowledge of the users.
Yeah, thank you. Thank you.
Hello, fantastic talk. So there are several approaches to deconvolute the spatial transcriptome. For example, I-star developed by Ming Li's lab can directly infer the single cell resolution RNA-seq by combining the spatial transcriptome and the image. And also Visium HD was released. So comparing this type of other approach, so your approach is consistent with them, or is there any difference?
Yeah, that's a great question. Thank you. So first of the I-star, if I remember correctly, that they more focusing on leveraging the imaging information for the high resolution imputation for for data, for example, that can facilitate some novel biological finding. But for us, first, the difference is that our method can integrate multiple tissue slices and then facilitate the domains that are detected. And second, our domains are defined by conceptually similar to the spatial niches that are characterized by different cellular environments and different cell type compositions. So that's, I think, the conceptually is also very different. And our domains is detected directly on these cell type informed low representations instead of the original transcriptomic features. And for the Visium HD, I think our method is scalable to the newest Visium HD dataset with millions of the spatial locations.